Probably all of you know about the structure and function of cell when you learn in your matriculations, plastostage, even lower class also. This is nothing but revision. বুঝতে পারলে? Okay. Any question? যদি কারো কোয়েশ্চেন থাকে সঙ্গে সঙ্গে জিজ্ঞেস করবে। তোমাদের আগের ব্যাচের অবস্থা কিন্তু খুব পোর আছে, একদম ভেরি পোর পারফরমেন্স। ইউ আর সেকেন্ড ব্যাচ, আই এক্সপেক্ট ইউ পারফর্ম ওয়েল। অ্যান্ড অ্যানাদার থিং, when we deliver lecture, everyone try to attend these classes. Do not converse with each other. Okay? Each class is very important. There is no repetition in the class. And MBBS is one of the most toughest syllabus in the entire world. Understand? समझ गए ना? उतना आसान नहीं डॉक्टर बनना। किसी को बचाना बहुत मुश्किल काम होता है। বুঝতে পারলে? মেরে দেওয়া বেরিয়ে যায়, গুলি মানে সঙ্গে সঙ্গে মরে যাবে, কিন্তু বাঁচানো ভেরি ডিফিকাল্ট। হ্যাঁ? So if you want to be a good doctor, knowledge is very important because knowledge is power. When I am working in a medical college, our teachers often utter one word: when the mind does not know, I cannot see. मन जा जाने ना चूकता देखे ना। बुझते पारले? समझ गए? नहीं समझे? अगर आपको कुछ नॉलेज नहीं है ना, आपको कुछ दिखेगा नहीं कुछ भी। इसलिए नॉलेज बहुत इंपॉर्टेंट है। So today's class is cell. This is a typical structure of cell. Everyone aware of this? Okay? There is two types of cells: prokaryotic cells and eukaryotic cells. We human being, mammalian species, all the upper species, they have eukaryotic cells. We have discussed only eukaryotic cells, not prokaryotic. So every cell has one surrounding that is called cell membrane. It is very important class. We are not going to discuss today cell membrane. Just listen the term cell membrane. There will be another separate lecture about cell membrane. So today we will discuss cell and the intercellular organelle. Okay? So this is a rough diagram of cell. You can see here this is cell membrane. Here the intercellular organelles. This is nucleus and some organelles. We are going to discuss one by one. Okay? So you need to wonder how the scientists they discovered this inside the cells. Anyone ask yourself? Yes, করেছো কোনো? কি করে এটা সম্ভব হয়েছে যারা? হ্যাঁ, কি করে? তারও আগে? This is quite recent advances. Electron microscope, even microscope also. জোরে বলো। রেড সেল দেখে? আচ্ছা, আর কিছু বলবে কেউ? আচ্ছা বলো তো, রেড সেলের মধ্যে একটা জিনিস নেই, এরকম টিপিক্যাল স্ট্রাকচার নেই রেড সেলের মধ্যে। রেড সেল হচ্ছে আরবিসি। আরবিসি তো বলছো? আরবিসি বুঝতে পারছি না। আচ্ছা, প্ল্যান্ট হয়েছিল। ওকে, we are going to discuss today only eukaryotic cells, not plant. Okay, this is the thing. You can see so what is the subcellular organelle? There are various organs inside the cell. That is called subcellular organelle. When the cell membrane is disrupted or dissolved by some solvent, Okay, like 2020 a liquid solvent, the organized particles in the cell are homogenized. That means they mix each other. They form a mixture. And this is usually carried out by 0.25 mole of sucrose as pH 7.5, 7.4. This is important. You may ask in the Bible exam. বুঝতে পারলে? আচ্ছা, before we proceed, everyone purchase your textbook Biochemistry. Satyanarayan, Vasudevan. Okay, very good textbook. You can follow any one of them, either Satyanarayan or Vasudevan. No problem. You can use anyone. But this is quite short period, now only 1 year. You have to read anatomy, physiology also. Then you have to follow one standard textbook like Satyanarayan or Vasudevan. Both are very good book. Satyanarayan or DM Vasudevan. Okay, so we continue our lecture. The organelles are then separated by applying differential centrifugal force. What is centrifugal force? Any idea what is centrifuge? Have you seen in laboratory centrifuge machine? দেখেছো সবাই centrifuge machine? কোনো আইডিয়া আছে? দেখে নিব কোনো? ল্যাবে আছে, দেখে নিও কি? ঠিক আছে, মেশিন রাখা হয়, জোর জোর ঘোরানো হয়, ঠিক আছে? It is called centrifuge machine. After the process of centrifugation, the molecule which are heavier, they settle in the bottom further side, and the lighter one they float on the top. Okay, you can see by this process we can separate the intercellular organelle. This is discovered by Albert Claudy, he got Nobel Prize in 1974. There is process called fractionation, subcellular organelle. Okay, and then we can see some markers. These are some enzyme markers. You may ask in the Bible exam. Please go through the textbook. I have written this, taken this from DM Vasudevan. Okay, first nucleus separated by C-700G for 10-minute centrifugation. Then in mitochondria 10 to 15,000 G for 10-minute centrifugation, and the marker is in an ATP synthase. This is one enzyme, ATP synthase enzyme. Okay, by this way you can separate lysosome, Golgi complex, microsome, cytoplasm. Cytoplasm is the most superintendent one, and the abundant enzyme is lactate dehydrogenase. Microsome enzyme is glucose 6 phosphatase. Okay, তোমরা যখন আগে যাবে একটু জানবে এগুলো, বুঝতে পারলে? This is from Vasudevan. Don't have to write anything, just follow the textbook. Vasudevan.
Sir, is it there in Satyanarayan?
Why not?
Every book.
If you দেখো, তোমার যদি জানতে চাও একটা বই ফলো করে চলবে না, তোমার মাল্টিপল বই কিন্তু ফলো করতে হবে, বুঝতে পারলে? কারণ সব বই সত্যি দেওয়া নেই কিন্তু। তোমার সত্যনারায়ণ ফলো করো, বাসুদেবন ফলো করো, ইভেন হারপার ফলো করো, ইভেন লেলিনজা ফলো করো, মানে প্রচুর মেডিকেল বই আছে বায়োকেমিস্ট্রি। हाँ पीपीटी, हाँ ले लो वहाँ से, कोई दिक्कत नहीं, वहाँ पे ले लो, मिल जाएगा। चलो, डोंट टॉक, प्लीज साइलेंस। नेक्स्ट organelle is nucleus. What is nucleus? This is the most important part of cell. Primary function of nucleus. কে বলতে হবে ভিতরে কি থাকে? নিউক্লিয়াসের ভিতরে খুব ইম্পর্টেন্ট একটা অর্গান থাকে, জিনিস থাকে, ক্রোমোজোম থাকে। হোয়াট ইজ ক্রোমোজোম? Very good. Chromosome is nothing but the DNA. They are folded, then packed in a very small area. Okay. With help of one protein that is called histone protein. Okay. Okay. We may ask in the Viva exam. Please tell me one type of cell or example of cell which do not have nucleus. Very good. Okay. Apart from RBC ছাড়া uppermost layer of skin cells. RBC do not have nucleus. RBC do not have mitochondria also. Okay. This is simple. Huh? I already read in class 12. Okay, nucleus surrounded by two membranes. The inner one is called perinuclear membrane with numerous pores. You can see here. This is inner membrane. The picture is not so clear. This is outer membrane. Here a lot of pores is there. Okay. With endoplasmic reticulum, see envelope this outer membrane with some ribosomes also attached there. Okay. This is called nucleolus. Okay. The outer membrane is continuous with membrane of endoplasmic reticulum. This is nucleus. Function is DNA synthesis, RNA synthesis, okay, protein synthesis. What is genetic material? What is gene? Any idea? Gene disease होता क्या है? Gene क्या है? What is gene? Very good. Functional segment of DNA which may produce protein. বুঝতে পারলে? DNA is nothing but a polymer of purine and pyrimidine. তোমরা এগুলো যখন পড়বে যখন পড়ানো হবে জানবে এগুলো, বুঝতে পারলে? দিস ইজ নিউক্লিয়াস। নেক্সট, নিউক্লিয়াস contains DNA, the chemical basis of genes which governs the function, all the function of the cell. What is nucleolus? In some portion of nucleus may be seen as lighter shaded area. This is called nucleus. And in the area of RNA processing, this is the area of RNA processing and ribosome synthesis. This is nucleolus. Nucleolus is very prominent in cells actively synthesizing protein. ঠিক আছে, যে সেল প্রোটিন তৈরি করছে অ্যাক্টিভলি, ওখানে নিউক্লিয়াস দেখতে পাওয়া যায়। দিস ইজ পোরশন অফ নিউক্লিয়াস। নেক্সট, what mitochondria? Any idea? ভেরি গুড। কি তৈরি করে ওখানে? এটিপি। কোথা থেকে তৈরি হয় এটিপি? কোন প্রসেসে তৈরি হয়? প্রসেস ইলেকট্রন ট্রান্সপোর্ট চেইন, অক্সিডেটিভ ফসফোরাইলেশন, নাম শুনেছো তো? তোমাদের এগুলো পড়ানো হবে। মাইটোকন্ড্রিয়াতে হয়। আচ্ছা, মাইটোকন্ড্রিয়া পাওয়া যায় না এমন কোনো সেল? আরবিসি। ভেরি গুড। ওকে, mitochondria is oval shaped or spherical shaped or rod like shaped, and they have 0.5 to 1 micron in diameter and up to 7 micron in length. The tail of spermatozoa is fully packed with mitochondria. Okay. Mitochondria also take part in some important function that is called apoptosis. What is apoptosis? This program cell death. Why it is so important? You know, cell has some life cycle. When the cell completes life cycle, you have to die. There is millions of cells dying every minute all of our body. So how they are dying? This is the process of called apoptosis, programmed cell death. Mitochondria's very important function is programmed cell death. This is called as apoptosis. Okay, mitochondria dysfunction is detected in chronic inflammatory diseases. This is structure of mitochondria. It has two layer, outer layer and inner layer. Okay, this is called matrix. This is called cristae. Okay. You may ask in the exam a short note of mitochondria. Anyone? Short note in nucleus, nucleolus. 5 minutes short note. First of all, brief introduction. What is this thing? Then you have to write one diagram with level diagram and some function. Each mark is very important. Each mark is count in your exam. Internal exam, internal assessment, even formative assessment also. Okay. If you do not pass in internal assessment, you are not allowed to sit in a final exam. Remember. So start your study from day one. It is quite difficult, I am telling you. If you attend in classes, lecture classes, and study well, it is very easy to pass, even good score. বুঝতে পারলে? না পড়লে তোমার কোনো ক্ষমতা নেই এসে পাস করানো। হয়তো তো আমি পাস করিয়ে দেবো, তোমরা কী ফিউচারে গিয়ে ডাক্তারি করতে পারবে না, ডিগ্রি পেয়ে যাবে কিন্তু কিছু লাভ কিছু হবে না। समझ गए ना? चलो नेक्स्ट we'll discuss mitochondria two membrane. Inner mitochondrial membrane contains enzyme of the electron transport chain. Fluid matrix contains the enzyme of cyclic acid cycle, urea cycle and heme synthesis. We will separate lecture classes in future about these things. Cyclic acid cycle, urea classes, heme synthesis. Okay. Next. These are some favorite questions in Viva exam. Remember. All the things are very important for Viva exam. Next. The function of mitochondria. First of all is very important, electron transport chain. There is generation of ATP. They part TC cycle held in mitochondria, beta oxidation of fatty acid. This is another process. We will read in lecture process, in spiritual lecture process. Ketone body production, urea synthesis partially some metabolism occurs in heme synthesis and gluconeogenesis and pyrimidine synthesis. Okay. So if you ask in an exam a short note, you have to write this thing. Okay. Any question? No? Next. Peroxisome. What is peroxisome? Jure bolo. What have I got? You write one what is mitochondria, then you have to write one diagram, then you have to write what the function of mitochondria. Okay, that's enough. Not everyone, so whatever you want, minimum 4-5 point তোমাকে লিখতে হবে. Next, what is peroxisome? These are some granular matrix. They are of 0.3 to 1.5 micron diameter. They contain oxidative enzymes. They perform various metabolic reactions. Have you heard the term oxidative stress? What happens if a bacteria comes inside your body? Not fever. Initially, what happens? Body try to kill the bacteria. বুঝতে পারলে? By the process of oxidative stress, oxidation, this body can handle this. Then. These granules have some very important enzymes. They are called peroxidases and catalases. They are all destructive enzymes. Peroxidases of polyunsaturated fatty acid in vivo lead to hydroperoxide formation. The free radicals damage nucleus, cell membrane, and tissue and gene. Okay. In future class we will let's discuss about this thing elaborately. Right now you can just see this. Inside our body, a lot of free radicals are producing by the process in this process. Catalase and peroxidases are the enzyme present in peroxisome which will destroy the unwanted peroxidases and other free radicals like hydrogen peroxide. Okay. In our body, sometimes hydrogen peroxide is generated by the process of metabolism. These are highly reactive. If you do not control them, they will damage your body. Okay. So these enzymes, they handle this peroxidases. Even the process of metabolism, lot of free radicals generated. Even the electron transport chain in the mitochondria, near about 1% of electron they escape from the electron transport chain and they form free radicals. They have to neutralized. Okay. Otherwise, they create a lot of problem. So peroxisomes are nowadays recognized as protective organ with an important role in prevention and genesis of neurodegenerative diseases and cancer. Like Alzheimer's disease. Have you heard the term? Alzheimer's disease? Patient tend to forget everything. Alzheimer's disease. Parkinson's disease. Patient tend to tremor, resting tremor. Parkinson's disease. These are all neurodegenerative diseases. They attack in brain. Okay.
So,
even the normal aging also. Alzheimer and dementia. Alzheimer is a term actually. Dementia is this also a dementia is what? Tend to forget. Alzheimer actually this is a part in the Alzheimer disease. Patient tend to forget everything.
at this age,
Almost same thing. Okay, motor neuron disease, about Ramdev, at the time motor neuron disease, amnesia, they tend to forget anything.
so all of them say.
Okay. Have you heard the name Stephen Hawking, one favorite scientist? He was suffering from one disease that is called amiable lateral sclerosis, ALS. Okay. ALS, amyotrophic lateral sclerosis, SCLEROSIS. This is also called as MND, motor neuron disease. There is progressive destruction of neural membrane. These are all neurodegenerative disease. Okay, the peroxisome, very important function, they tend to prevent these diseases. These are some diseases that can occur. Deficiency of the peroxisome matrix protein can lead to this adrenoleukodystrophy. You will read in future medicine lecture classes. Okay. This is characterized by progressive degeneration of liver, kidney, and brain. What is degeneration? This simply you can say aging. বুঝতে পারলে বুড়ো হয়ে যাওয়া ডিজেনারেশন. This is a rare autosomal recessive condition. We will read in lecture class in future. The defect is due to insufficient oxidation of very long chain fatty acid by peroxisomes. Some of this is like Zellweger syndrome. This is quite difficult term, right now for you. The protein are not transported into the peroxisomes. This leads to formation of empty peroxisomes or peroxismal ghosts inside the cell. Protein targeting defect are describing something I taken from Vasudevi. You read there. Okay, just remember these three terms: adrenoleukodystrophy, Zellweger syndrome, and primary hyperoxaluria. Okay, these are some clinical conditions and defect in peroxisomes. Okay, next is Endoplasmic Reticulum. Okay, what are they? They are some network of interconnecting membranes enclosing channels or cisternae that are continuous from outer nucleus envelope to outer plasma membrane. Nucleus to plasma membrane. There are some interconnecting membranes are there. They called endoplasmic reticulum. Some reticulum they attached with granules. They call rough. Some are not granules, smooth. Granules are ribosome. Function is what function? Their primary function is to fold protein. The function কি আছে? Here function কি আছে? To fold protein. In the process of translation from messenger RNA, protein is forming. Yes, in a long chain. These protein are not useful. They have to go their good shape to 3D structure. This is called post-translational modification of protein. This occur in ER. Apart from this, detoxification of various drugs, very important function of ER. Microsomal cytochrome P450 system. They are responsible for a lot of drug metabolism. বুঝতে পারলে বা ফার্মাতে এগুলো পড়বে even in our lecture class in xenobiotics lecture class there they will discuss these things elaborately. Example of some drug like morphine, phenobarbital, these are drugs they metabolize inside this system, microcephal cytochromal P450 system. What is ER stress? The protein that they are generating inside our body, they coming inside the ER and they getting their shape. When the number of protein are increases to the more than this capacity, then is called ER stress. Today everyone should go to their textbook, read this chapter. Vasudevana Satyanarayana, any book. Then you can understand what is endoplasmic reticular stress. One of the most common disease where the pathogenesis involved in ER stress is diabetes mellitus. What is diabetes mellitus? কি বলো কোনো আইডিয়া আছে ডায়াবেটিস কি নাম তো সবাই শুনেছো ডায়াবেটিস হোয়াট ইজ ডায়াবেটিস ইনসুলিন রেসিস্ট্যান্স ওকে আর কিছু গ্যাংগ্রিন পরে আগে হয় না এটা কমপ্লিকেশন Diabetes mellitus is a metabolic disease characterized by hyperglycemia, glycosuria, ঠিক আছে polyuria, polydipthia, বুঝতে পারলে समझ गया gangrene this is a complication of diabetes complication long term complication ঠিক আছে long term complication হয় ডায়াবেটিক রেটিনোপ্যাথি হতে পারে চোখের মধ্যে হয় রেটিনোপ্যাথি নার্ভে হয় নিউরোপ্যাথি ঠিক আছে কিডনিতে হয় নেফ্রোপ্যাথি ঠিক আছে আর কি হয় পেরিফেরাল ভাস্কুলার ডিজিজেস there is a complication of peripheral vascular disease gangrene atherosclerosis edema কেন হয় edema হয় না ঠিক আছে next is lysosome. Lysosomes are tiny organelle. Lysosomes have acid pH and most of the enzymes are acid hydrolysis. Okay. Endocytic vesicles and phagosomes are fused with lysosome to form the secondary lysosome and digestive vacuole. When a bacteria attack your body, some cells like neutrophils or macrophages they engulf them. That is called phagocytosis. And then a vacuum is created. Inside the vacuum there is bacteria, and then this vacuum will attack with lysosome, and then this enzyme destroy the bacteria. Okay. So very important. Even when the cell is dead in the process of apoptosis, sometimes this lysosome also use. As long as the lysosome membrane is intact, the encapsulated enzymes can act only locally. But when the membrane is disrupted, the release enzyme can hydrolyze external substrates, leading to tissue damage. Say example, myocardial infarction. What is myocardial infarction? There is blockage of coronary artery by sometimes like plaque or infarction, anything. Then this area supplying this blood, they died. And then this enzyme can coming out from the cell, and this enzyme, lysosome enzyme, destroy everything. They makes up everything out. Lysosome. So this is say this is bacteria engulfed by this cell. Say this is example of neutrophil. So lymphocyte, okay, nucleus, because neutrophil has multiple lobes. Okay. Then this phagosome, they attach with these lysosomes with enzyme is there, and they form lysosome phagosome fusion. Okay, this is called secondary lysosome, and then bacteria they killed and they become debris, and these debris is go out, and the enzyme is again go back on the inside the cell for reuse. Okay, this is lysosome. Clinical application of lysosome. Gout. What is gout? Very good. There is increased number of uric acid in the blood. Then uric acid tend to precipitate in the peripheral joint, where the blood circulation is less, temperature is less. Once they precipitate, this enzyme come and they engulf them and try to kill them, destroy them. And as a result, what is this? They form lot of intense inflammation in this area. There is increased pain, swelling. This is a very bad condition. Any patient suffer from gout, they can tell you how dangerous it is. Pain is so severe. Gout pain. Next, following cell death, lysosomes rupture release the hydrolytic enzyme, which bring about postmortem autolysis. Okay, lysosome can destroy the surrounding cells. Lysis means to burst out everything. Auto means self. Certain cancer cells liberate the cathepsin, this lysosomal proteases, one enzyme, out of the cells. Then this cathepsin degrade the basal lamina by hydrolyzing collagen and elastin. Okay, there is some, you can say, rope-like structure. They held them in a local area. If they destroy, then this cell will release in the circulation, cancer cell, and they try to start traveling and settle some other places. This is called metastasis. Okay. There are some few genetic diseases where lysosome enzymes are deficient or absent. This lead to accumulation of lipids. There is some example of clinical utilization of lysosome. Some silicosis. What is silicosis? The person living in a highly polluted area. Okay, lot of dust is there. The silica is there. Silica they inhale, and then this is silica they try to kill by this enzyme. They form a mass-like structure inside the lungs. This is called silicosis. Silicosis. Inclusion cells disease, also known as mucolipidosis 2, is a rare and fatal genetic lysosomal storage disease. The condition gets its name from the inclusion bodies or dark inclusions of undigested materials that build up inside the lysosomes of mesenchymal cells like fibroblast. Okay. So you may put on short note inclusion cells. How this occurs? Defective enzymes, failed enzyme trafficking, enzyme leakage, and cellular accumulation. Golgi apparatus. Golgi organelle is a network of flattened smooth membrane and vesicles. It may be considered as the covering of endoplasmic reticulum. The main function of Golgi apparatus is protein sorting, packaging, and secretion. Okay. They may pass through the plasma membrane to the surrounding medium. They reach plasma membrane and form an integral part of it, but not secreted. They are formed into the secreted vesicles, where these products are stored for a longer time. Okay, under appropriate stimuli, the content are secreted. Example: release of trypsinogen by pancreatic acinar cells and release of insulin by beta cells and Langerhans cells. Okay. Sometimes you can see, say example, this is a cell, the nucleus, they are producing protein and they are stored here. Okay. Then when required, they will move into the cell membrane, then release. They are called Golgi apparatus. Any question? আপনার বাকি পড়ে না, বলে যাবে কিন্তু সব বলে যাবে এবং যে কোনো একটা কোয়েশ্চন আসতে পারে, বলে দিচ্ছে শর্ট में আসতে पारे. समझ गए? नहीं समझे? यहाँ से कुछ भी क्वेश्चन आ सकता है. शॉर्ट नोट 5 समय का आ सकता है. साइटोप्लाज्म और साइटोप्लाज्म. You can see this is a matrix of a cell, J-like structure. Everything they are inside this cell floating in the cytoplasm. Okay, this is a living material. Lot of enzymes they are in cytoplasm. Say example of enzyme in glycolysis. Okay, lot of our reactions enzyme is present in cytoplasm. In eukaryotic cell, cytoplasm is everything within the cell membrane but outside the nucleus. Prokaryotic cells with lack of membrane bound nucleus, the cytoplasm contains all the cell's contents including genetic material. I will not concern with this thing, just forget this. Cytoplasm composed of 3 main parts: cytosol, organelle. This is nothing but semi-fluid, gelatinous portion of the cytoplasm, primarily made of water, salt, and various organic molecules. Okay, protein is there in cytoplasm. Inside the cell, protein are electronegative. Remember, organs are there: mitochondria, endoplasmic reticulum, Golgi apparatus, ribosome. All are present in cytoplasm. What is cytoplasmic inclusions? Insoluble particles suspended in the cytoplasm that are act as storage sites. These can include reserve foods like starch and glycogen, lipid droplets, and pigment granules. Cytoplasm, a network of protein filaments and tubules, they provide structural support and maintain the cell's shape. It is also involved in the movement of material within the cell. Okay, function of cytoplasm: structural support, metabolic process like glycolysis, protein synthesis, fatty acid synthesis, transport of material, okay, like nutrients, waste product, they transport, storage, some nutrients, ion and enzymes, they are stored in cytoplasm, and there is protection. Its gel-like nature acts as buffer, protecting the cells organelle from damage caused by external forces. Okay, difference between cytoplasm and cytosol. Cytoplasm means this is a comprehensive term that includes the cytosol, all cellular organelle and cytoskeletal and inclusions. Cytosol refers to specifically to the jelly-like fluid component of the cytoplasm. Okay, you may ask in the bio exam, what is the cytoplasm, what is cytosol? Okay, so this is just today our topic. So thank you very much for listening.